Well, first I'd like to thank all the people who have organized my visit here for inviting me to come to this part of Spain, which I never visited before. And I have to say that I've had, so far, a very good time. But unfortunately, it's been too much eating, too much drinking, too much talking. So now I have to talk again, because as you know, you do not get food till you sing for it. So I'm going to sing for my supper. I've been asked to give a lecture on what's called the human genome for an audience that is not professional, does not know any genetics. So I have to start at the very beginning. So of course, all the professional people should forgive me for making things perhaps a little too simple. So what is the most important biological observation that we know of must have been the first thing that an intelligent man noticed when he walked on the surface of this planet, is that animals and plants reproduce, but they always reproduce their own kind. As we say, like begets like. So flies give rise to flies, humans to humans, sunflowers to sunflowers, and that must have struck everybody because, of course, they were observers of nature. So how is it, you might ask, that a complicated organism like an elephant can produce another elephant? Because clearly what passes between the generations is not the elephant itself, although for a long time they thought there was a little homunculus in the sperm, and all it did was penetrate the egg and then grow big. We now know it's not the object itself, but a description of the object. A description, I don't call it a program, I call it a description of the object that is handed down from generation to generation. Right? Now, what is this description? What is, if you like, the information that we hand down? The remarkable thing is that in 1953, for the first time, we could actually study information in the living world, because information is coded in chemistry. So an information molecule is the DNA sequence that lies at the heart of what you pass down. And so we had another quantity in biology. Everybody was concerned with matter. How did biological organisms make all the things inside them? Or with energy, where did they get the energy to do this? And the revolution that we had in 1953 is that we have to deal with a new concept, and a concept that is embodied directly in the sequence of DNA. And once you have that, then I think we can now start to do real science on this problem, which I think could not be done until we had the structure of DNA. Because anybody who saw it, and I saw it as a young man very early, before it was published, 1953, and I just said it was a complete revelation, because now I knew what had to be done in order that we could understand how living creatures reproduced themselves, how they copied the information. But I'm going to tell you a little story which is not widely known. A very famous physicist and mathematician called von Neumann. And von Neumann got very interested in just the abstract question of a self-reproducing machine. He said, how would I make a machine that would copy itself and reproduce itself? And he then proved, it's a logical proof, that you had to do it in the following way. First, you had to have a description of the machine. That description, which we can call a tape, which in fact we would call a gene or DNA, that description is then used by the machine to build another copy of the machine. And then at the end, you have to copy the tape and insert the tape in the next machine. And if you look at that, that is exactly in the logical form of a self-reproducing machine, exactly what the DNA accomplishes biochemically in the exact state. So it is an implementation of that. Now, of course, although I have to tell you that von Neumann had these thoughts, and he wrote about it in his theory of self-reproducing machines long before people knew about DNA. But such was the culture of science that nobody knew about von Neumann, including people who it is very clear actually were an audience at a lecture he gave in California in 1951, long before this. So that is how history looks as though von Neumann is the genius behind everything. But if you ask Watson and you ask Crick, they wouldn't have known who von Neumann was. Of course, von Neumann is very famous today for not only inventing, but he invented the modern computer. He didn't actually build it, but he invented the form of it. And what he said, you could represent this in what's called a von Neumann machine, because you can have a tape which you read, and of course that dates back to an earlier discovery by a man called Turing in the 1930s. That is, what can be accomplished in any machine that can be expressed in terms of a universal Turing machine, because it says it provides you with the mechanical rules for computation of the answer. Well, we must now try to find out about this tape, this tape within us, and let's just look at what it is. So it consists not of these blocks, but I mean of these letters, which represent the individual chemical entities. It's a language called of nucleotides. There are 4 kinds, called guanine, cytosine, adenine, and thymine. And there is a principle of self-complementarity here, so that it doesn't consist of one sequence, it's 2 sequences which are paired according to the rule wherever there's an A, there's a T in the opposite strand, and of course wherever there's a T, there's an A, and wherever there's a G, there's a C. So given any sequence of DNA, you can write down the opposing sequence. But the beauty is, if you separate the 2 sequences and assemble the complements on either side, you land up with 2 copies of the double-stranded DNA. And that's actually how it occurs in nature, that we copy the strand into one. And then, of course, that says how we can copy it equally. So we can copy the tape. The tape can now go and make a new machine under these instructions that are written on the tape. So in a sense, the whole of the living world provides a fascinating and extremely deep question about a number of things. One, how did this complexity evolve? How does it that we have such wide variation? And then how is the tape read? Because the tape is read in order to make real material objects. So we know that a lot of it is translated into another language. It's a language, another chemical language, except this language has 20 different things called amino acids. And each of the tape is simply translated into this. And so one of the fundamental questions that we asked right at the beginning was, how do you go from a 4-letter language to a 20-letter language? And many things were done, but of course, we now know that there's a code, that 3 letters in the nucleic acid language stand for 1 letter, that there are multiple representations for some of the amino acids. And the remarkable thing is that to a very large extent, the code is the same in all branches of life, whether they are viruses or bacteria or fish or people, we all have the same genetic code. So there is a unity of chain. Now, of course, when you have a code like this and you are copying it, you can make errors because, as everybody knows, nothing is perfect. And so when you make errors, you can put in the wrong base. It can be done with certain degrees of mistakes, and of course we call those mistakes mutations. And if the mutation is in certain bases, we can change one amino acid into another. So we can change the product. So we can make alterations. And just to give you some idea of what it is, there are today, I'm told, 7 billion people, 7 times 10 to the 9 people in the world. Okay? There are 6.4, I think, billion bases in the 2 copies of your chromosomes, because you get 1 copy from your father, 1 from your mother. So we have that. That means almost certainly somewhere in the world, every mutation has already occurred. Of course, we can't see the effect because they have 2 copies. Right? So this means that if we could find everything, we could explore the whole potential changes that we could do by doing this. And of course, since mutations could occur and were noticed in nature, and of course we can induce them at a higher frequency with various forms of radiation or chemicals, we could actually change organisms. We could make mutants. And that began the subject of genetics, which Mendel did in the middle of the 19th century. He started to cross peas, and he had a very simple theory that how these genes, they were called Mendelian factors, and they gave rise to characteristics of the organism. Today, we would say the Mendelian factors are called genes, and the collection of them is called the genotype, and all of the DNA is called the genome. Those are just words. And of course, what they present themselves is called the phenotype. That is what the genes do. Right? And if genes can make proteins to do the work that is in the cell, and so all catalysis throughout the universe of life is carried out by these proteins. And they are remarkable, flexible things. They can do many things. And we now know of hundreds of thousands of different proteins. We seem to find many collections. Every time we explore another feature of the living world, we find curious organisms that can live, for example, in boiling water. There are bacteria that can live on hydrogen. And the most fascinating one is if you go along any highway, you will find bacteria in the soil next to the motor roads that can actually deal with carbon monoxide. They can actually use carbon monoxide and live with it. That's, of course, what's come out of the exhaust of cars and has enriched the growth of these bacteria that, of course, are hungry for many things, for many carbon compounds. So we have all of this in this vast armamentarium. For years then, geneticists tried to explore the relationship of the genetic material, which they only knew were on chromosomes. They could try and do this by genetic mapping. So those were all the great accomplishments of the classical experience of genetics. And they tried to understand how it is that you make a hand, right? And of course, we don't have an instructions to make a hand in the same way, because if you were to ask someone, how do you make a hand? He'd probably write a computer program saying for n equals 1 to 5, repeat, make a finger. So you make 5 fingers, and then you make a hand. We don't make hands that way. So the discovery of how all of this construction has now occupies the people in the life sciences. Right? So what could we find out? Could we short-circuit everything by actually finding out what is written in the DNA? If I had the sequence of bases, which of course we tried to get by genetics in the old days, could one determine the chemical composition of the DNA? This is just a problem in chemical analysis. We call it a sequence because these are very long, very thin, one-dimensional organisms. All the DNA in your chromosome is one molecule. It just happens to be a very big one, a very long one. You have about 1 meter of DNA. The spacing between the bases is 3.3 angstrom units. So you have 3 bases per nanometer. So it really is nanotechnology. In fact, it's sub-nanotechnology. And that is quite an interesting way of looking at it. Right, so, well, Fred Sanger, he invented a way of sequencing proteins. Up to the time he did it, most chemists did not believe that proteins had a formula. They thought they were just sort of messes, you know, globs and polymers of some undrawn. And I was at a meeting in the 1950s where the doyen of British chemists, Robert Robinson, after Fred Sanger gave his talk on finding the sequence of insulin, which was the first one he did, and said, this is remarkable. It means that proteins have a chemical formula. Like I can write down the formula for urea or for any one of the chemicals that we were doing. You can write down the formula for a protein. You could also write down the formula for a nucleic acid. So happens to be called a sequence, but please, it is a formula. And you can synthesize it if you want to. People have synthesized protein. Today, people have synthesized DNA. That's called now, a new name is called synthetic biology, but it's not, because all they're making are forgeries. They copy another piece of DNA and they put it inside a bacterium, which means that they have forged it. They haven't invented anything. They've made a copy, and of course they're not very good counterfeiters because they put their names on this as well, so we can catch them very easily and say, that's not a real gene. I mean, that's just a copy of a gene. And so it's a counterfeit. It's not the real thing. However, well, so now there is this following challenge that we can actually decompose the huge molecules, the huge complex genomes, and we can find the sequence of them. And when the first organism, you have to realize that was completely sequenced of any complexity was a small virus for bacteria called lambda. It had 45,000 bases, 45 kilobases as we call it. And at that time people said, what if we could sequence the human genome? Well, of course, that was a tremendously huge jump in magnitude from 5 times 10 to the 4 bases to 3 times 10 to the 9. Yeah, 5 orders of magnitude, just like that. And of course, it was a challenge, and people then decided that this is what they wanted to do. But first they tried smaller things like flies and so on, and gradually we worked up the scale till the genome became possible to sequence. Now, of course, there are 2 ways of doing this sequence. One is what I call 3M. 3M is a famous company in America. For us, it stands for machines, money, and management. That is, you can set up a big lab, and I, in fact, once suggested that we should set up a penal lab so that everybody caught cheating, copying other people's work, would be sentenced to this lab. It would be in a very undesirable place like Chicago, and he would be sentenced to do 1 megabase, that is 1 million bases of sequencing. But that was just a joke, of course, but was taken quite seriously by some people. So eventually, by it was calculated that it would cost about the same as the number of bases in dollars. So it would cost about $3 billion, which today sounds trivial, peanuts compared to the trillions that people are talking about. As I read a book by one of the people who was the treasurer, he was a treasury secretary in the last Labour government in Britain, and he was worried because he had to produce something like £5 million to save a bank. He said little did he know that in a few months he would be writing checks for £50 billion, and these numbers now don't mean anything, few trillion here and there. I mean, what does it matter? So, but these were real numbers. You had to do it. So, of course, it required either some new advance in technology, so it could be done very simply, or it required some unorganized activity. It also required raising a lot of money. And so it became very similar in the way people saw it as to put a man on the moon. Also cost about $3 billion. And they decided we would like to do this by the end of the century. That is, by the year 2000, we should do this. Now, in a curious way, it was very similar to putting a man on the moon. Because if you think about it, it's very easy to send a man to the moon. That's trivial. The problem is, how do you get him back again? And that's the expensive part, is to get him back. So in a sense, it was very trivial to sequence the human genome, which is now, so to speak, stranded on a moon there. And now we have to find out how to get it back. We have to interpret it. we all knew that that was going to be the biggest task of all, because we had no experience in encountering such a mass of information. Well, as many of you know, when it was sequenced, as I used to say, when Tony Blair and Clinton announced they had sequenced the human DNA between the two of them, and that it was all done, they left the next job to us scientists. But everybody said, this will be the great message that they can bring to everybody. We will have new drugs, we will solve all diseases, we will solve all human problems. And that was, I think, as they say in the trade, it was a bit of hype. That is, it was exaggerated, because there was nothing like that in the end. And the 10 years was celebrated this year, actually the end of last year. And people are saying, well, what actually did you deliver for this? And we have to say, not much. But one of the things is we just had no idea, and there still is not a good idea, not good ideas on how to deal with the interpretation of this information. So the first concept I'd like to tell you is the concept of junk, all right? And I must just define that very carefully. See? Because if we said every base in your genome is important, which it is in a bacterium, because every base counts, if it was important, how many genes would you have? So we began to think, well, how many genes does it take to make a complex thing like a human? And what we knew is we had sequenced bacteria. In fact, the common bacteria of your intestine, E. coli, it's got 4,000 genes. So everybody thought, well, I'm much more complicated than E. coli. I must have many more genes than E. coli, right? And so the guesses must have hundreds, thousands. But if your genes were the same as E. coli, we can make a calculation about how many genes you might have had. There's something called, it's a short gene, it makes an average protein. Give or take, we would say, you must have, if they were E. coli-like genes, you must have 3 million genes. That sort of sounded okay, I mean. But then, of course, we could find other genomes which weren't so big. And in particular, by looking carefully throughout nature, we found genomes which give rise to quite complicated organisms called fish that share a lot with us. They have the same genes as we do, and they had 10 times less DNA. So were you going to argue that a fish has 10 times fewer genes than you as a human? And it then became very clear. Of course, you could argue that. You could say, I'm much better looking than a fish. I must have genes more complex. So we've got to a stage, I think you can see, where we're beginning to wonder, what do we mean by complexity? And what is this measure of complexity that we've got to deal with? And do we mean that because I'm much more complicated than a fish, I just have to have lots and lots of more complex genes? It's not correct, because it is made up of actually processes that are gene assemblies, that is, where proteins work together. And therefore, by doing this, they're like, if you like, subprograms in a text there. You don't have to represent everything at the common thing. And of course, we used to speak very loosely of a gene for this and a gene for that, to speak for a gene for blue eyes. And we now know that has got nothing to do with blue eyes. It's got to do with a chemical process that moves certain amino acids across membranes. So that's the real representation. Well, in order to try and explain this in more detail, let me consider the following argument. Right? You know, we have our first cousins are chimpanzees. And our first cousins, we have genomes that are very similar to ours. 99% is the same. We only differ in 1% of the DNA. So everybody immediately could argue, when we sequence the human genome and we sequence the chimpanzee genome, we will find the human has one extra gene. And that gene will be the gene for language. In other words, we're going to represent all of language in one gene. All right? You can see that this is not a correct argument, because I always used to have an alternative argument for this. For example, suppose that chimpanzees during their history found that talking gets you into trouble. They would have evolved a language suppressor gene. In other words, the language would have been a natural process, and they would have suppressed it. And of course, they would have the exact gene. So whereas, according to the one model, we would have the one gene for language, which we would call the Chomsky gene in man, according to the Brenner theory, it would be called the Chomsky gene, because they would have the extra ones. So, in fact, in order to talk about function, you have to know what you're talking about. And I think that this is still very, very badly understood by people. They think there are genes for schizophrenia, genes for cancer, genes for homosexuality. But you could also ask, maybe there are genes for luck. Maybe you can have good fortune because you've got... It's not represented like that at all. So we must really get to grips now with what the genes do within you. Well, that's the first thing. So we need another definition of a gene. It hasn't got to stand for a character, abstract character like this, but it has to stand for a process of some sort that is, in the end, chemical. That's what happens inside your body. Could be an enzyme, could be a transport program, and that is displayed in the real world in terms of a complex function that you can perform. And that is the task that we really have to have and do. Now, of course, most people are interested in the question, not so much of the fundamental biology, which I just touch on for the moment, but much more is, how's it going to affect me? How's it going to affect my health? Is it true now that we know all of this, we'll be able to cure any disease and do anything like that? And the answer is, it's still a very long way from that. And the reason for that is we've got completely the wrong perspective about the human genome. If you were a biological entity, you are at the mercy, so to speak, of your environment. If you're a dog or a cat or something like that, an animal, a plant, you're at the mercy of your environment. If your environment changes and you can't actually adapt to that change yourself to adapt to that change, then you become extinguished, you become extinct, and nature just tries again with something else. So imagine we humans at a time when the world was freezing, the ice ages were coming on. In order to survive properly under those conditions, they could only do it, as many of the animals did, by growing hair, by growing a coat that they could wear. But humans didn't have to wait for that. They had another tool, which is the brain, and what they did is they went out and killed an animal and put on its skin. There you are. Huge process that would take thousands and thousands of years of biological evolution to try to grow hair on your body to keep warm. They just did it overnight by taking it from the animal, right? And so for the first time, and I give that as one example, there are many examples like this, because we could cook food, actually, because we discovered fire. We could actually cook food, and we could actually make things palatable that would be impossible for us to eat unless we had very special adaptations to this. So we could widen our diet. We became omnivorous. And by this second process, which we can call cultural evolution, man then came to change his environment. He didn't like it. If it was dry, he said, I'll bring water here. And of course, that is now the history of humanity as we know it. But biologically, humans are still the animals they used to be before their brains got bigger. They're still the animals, and what our body has adapted to is the following. Just to give you one example. The body says, whenever you get food, eat as much as you can. Eat everything, and then take the food, convert it into fat, and store it. Because it turns out fat is the most efficient way of storing energy that you can use later when there is famine or the animals disappear. It means you will have prolonged survival. So our body developed purely biologically, certain parts of our brain, a internal state that actually contributed to our adaptation and survival in conditions that now no longer exist, because we can change them. So if you like, we've got inside of us a genome which is probably several 100,000 years old, evolved under the conditions of the planet several 100,000 years ago, with no supermarkets, no cars, everything like that. We got all this biology, and now we're living in an environment to which it is completely maladapted. And I think that is the critical understanding that we have to have about our condition in the kind of culture that we live in now. And in fact, this is the whole history of humanity, which we need to understand fully, from how they changed from being small bands of people that were hunting or collecting things to then this organized society that we have now. And as you know, these things then change very rapidly. And of course, we can now begin to see what is the most common disease of culture. It is the epidemic of obesity, And this means that the number of people who have type 2 diabetes is rising. It's rising fast in China as they begin to live in a society which has different conditions, and it's rising fast in India. And it is just inevitable. It is the history of our own that's now not adapting to this.